All right, thank you very much. If you do the next slide, please. Thank you very much. Today, I want to speak on behalf of USDA and Food Safety Inspection Service, and I'll provide an overview of FSIS's role in the regulation and microbial biotechnology used in meat, poultry, and egg products. Our regulatory process is essentially the same, whether it's a traditional or typical substance like a food ingredient or a substance derived from food microbial biotechnology. As such, the mission of FSIS is to ensure that meat, poultry, and egg products are safe and that they are properly labeled and packaged. Our authority includes these acts, including Federal Meat Inspection Act, Agricultural Marketing Act, Poultry Products Inspection Act, Humane Methods of Slaughter Act, and the Eggs Product Inspection Act. Next slide, please. Our role in the regulation of microbial biotechnology, as I said, is the same as traditional, and it will involve the review and approval of new technologies, including food ingredients derived from microbial biotechnologies, as well as the labeling of bioengineered or genetically modified ingredients. Next slide, please. All right, the next few slides, I'll describe our new technology review process. Next slide. Okay, our new technology is any new or new application of equipment, substances, including food ingredients, methods, processes, or procedures affecting the slaughter of livestock and poultry, or the processing of meat, poultry, and egg products. Food ingredients are substances added to food contact substances, including packaging components, and generally recognized as safe or GRAS substances. Now, most new technologies submissions generally include processing aids and ingredients such as antimicrobials, binders, acidifiers, alkalizers, and flavoring agents. And likely food ingredients or substances that might be derived from microbial biotechnology would include genetically engineered microorganisms, bioengineered or genetically modified ingredients, or substances that might be derived from microbial biotechnology. Next slide. Now, under the Federal Meat Inspection Act, the Poultry Products Inspection Act, and the Eggs Products Inspection Act, FSIS is responsible for determining the efficacy and suitability of food ingredients in meat, poultry, and egg products. Now, suitability relates to the effectiveness of the ingredient in performing the intended purpose of use and the assurance that the conditions of use will not result in adulterated product or one that misleads the consumer. Now, in particular, suitability data, this would support that neither the intended use nor the substance would adversely affect the wholesomeness of the product. And this may include data that illustrates how the technology affects the organoleptic properties, such as taste, color, odor, and texture of the product. Next slide. All right, we evaluate the technology to determine essentially 5 different areas: whether it affects product safety, whether it violates FSIS regulations, whether it interferes with inspection procedures, whether it jeopardizes the safety of our inspection program personnel, or is it deemed suitable for use in meat, poultry, and egg products. Next slide. Now, this slide gives an overview of the new technology review process. Let me just put this in here. Now, under our process, there are 5 main steps that are involved in the process. And this includes our subject matter experts from various offices within FSIS. And what we would do is companies would submit scientific data or technical information that describes the technology or a new method, process, procedure, and what the technology is intended to accomplish, as well as the beneficial or adverse effects that the technology is expected to have on FSIS regulated products. Now, this process involves 5 steps, which would include the submission would be received by the Office of Policy and Program Development, the Risk Management Innovation Staff. And this is usually through our, we have a computer program system called FSA Ask or email, and you would log into our new technology management system, and your request would be assigned to a project manager. Now that project manager would do an initial review to check for completeness, and it would contact the company if there's anything needed additionally. That project manager will set up what we call our technical review team and allow them to review the submission. Now, after concurrence is reached by all members of the technical review team, the project manager will draft and clear the official correspondence, which we call a no objection letter. And in most cases, the Risk Management and Innovation Staff Director will sign that letter, and that letter will be emailed to the submitter. Now, each new technology submission is reviewed by a multidisciplinary team that are tasked with evaluating the scientific or technical support. And this review team, members are selected based on their expertise and from varying staffs from several offices, primarily Office of Field Operations, Office of Public Health Science, Office of Management, and Office of Planning, Analysis, and Risk Management. Next slide. Now, there's some key points that I want to point out about our protocols. They should include the following information. The intended use in summary, and this would be a detailed description of what the technology is intended to accomplish, as well as the beneficial or adverse effect that the technology is expected to have on the products. The method of use, and this describes how the technology will be used, including the method of application and operational parameters where applicable. It also provides the ranges for the operation parameters specific to the intended use. And just an example, for all applications, this could provide, we'd ask that the company would provide the concentration, such as the minimum and maximum safe, for instance, if it's a pH, in the form of a range. And if safe for spray application, we would ask that you include the minimum and maximum contact time and pressure in the form of a range. Now, we request this info to be provided, for establishments, the most comprehensive information possible, so that the substance can be used appropriately to maximize its effective use. Now, in addition, this mitigates any confusion for our inspection personnel, program personnel, regarding the use of any chemistries of the establishment. We also ask for monitoring, in terms of monitoring and evaluation, that you describe the technology, how it will be monitored and verified. This will include what parameters should be monitored and at what frequency, and also includes the explanation of how records will be maintained. Now, very important for us is the submission of the safety data sheets, and this concerns the safety of our inspectors. Now, for the safety data sheets, we ask that you include the use concentrations of the substance and include a description of how the use of the technology would not adversely affect the safety of our inspectors. Now, our workplace safety and health hazards include inhalation, direct skin contact, and fire and explosion caused by exposure to the product. Now, it's working solution or dry mixture and potential byproducts formed, and maybe if there's off gas during the application process. Now, this employee safety information should demonstrate how the new technology will not jeopardize the safety of our inspection personnel, program personnel. And this is described in the FSS compliance guideline procedures for new technology notification and protocols, which is on our website. In addition to the safety data sheet, we ask that you would address certain information in regard to employee safety. And One of the main concerns we have is potential workplace exposure. And this should be addressed and include that what could result from inhalation and direct contact during this use of the substance technology. If we do not currently permit our inspecting program personnel to work in conditions where respiratory protection, eye goggles, or clothing that can protect against severely corrosive materials is required. And this should be discussed in the provided safety data sheet. Now, if there are no workplace exposures expected and no control measures needed, we ask the submitter to state this and provide the reason why. And we please also ask that you indicate how our inspectors be informed of the above information so that it is clear and that the information contained in the safety data sheet for the concentrate does not apply to the actual workplace. Next slide. This slide provides some of the contact information and online resources. And as I mentioned before, our AskFS, which is a web-based computer application and phone system, is designed to receive and respond to technical and policy-related questions from our inspectors, our industry, and other stakeholders. And new technology submissions can be submitted to us through AskFS. And also, I just want to point out the guidance document that I described can be found on the new technology webpage. Next slide, please. I do want to mention one caveat. Now, in accordance with the Memorandum of Understanding between FDA and FSAS, FDA is responsible for determining the safety of substances, including GRAS substances, food additives, and color additives, as well as prescribing safe conditions of use. Now, as previously discussed under our Federal Meat Inspection Act, the Poultry Products Inspection Act, and the Eggs Product Inspection Act, FSAS is responsible for determining the efficacy and suitability of food ingredients in meat, poultry, and egg products. Now, it would be prudent for establishments or companies that manufacture new substances or design new uses of previously approved substances, including any changes in the concentration or application of methods, to first submit their notification to FDA for safety determination before submitting it to FSAS. Next slide. Now, the next few slides, I do want to go through some details on the labeling of bioengineered or genetically modified ingredients. Next slide. Okay. Agriculture Marketing Service has jurisdiction over bioengineered disclosure, and more information on that can be found on the AMS Industry Fact Sheet at this website, and also list the website where you can ask further questions, or you can call the phone number that's on the slide here. Now, we do consult with AMS on the requirements of the rule, and we are evaluating questions raised by industry that are related to our products. Next slide. Now, in terms of labeling of National Bioengineered Food Disclosure Standard, FSAS is requiring that companies, obtain one sketch approval for each BE disclosure format used, and that subsequent labels using the previous approved disclosure may be approved generically. And this website provides more information on our label approval of BE disclosures. The labeling record should include documentation to show compliance with 7 CFR 66.302. And this is the record keeping requirement. And here are some examples of records. But I do want to point out some additional information regarding this stuff. A statement about BE, a bioengineered food or bioengineered food ingredients made in compliance with the standard is considered a special statement or claim that requires approval for compliance in accordance with 9 CFR 412.1E. Establishments required to make mandatory bioengineered disclosures in accordance with the standard will need to submit at least one label containing the disclosure to our labeling and program delivery staff for prior approval. and subsequent similar labels that use the same label disclosure and format under the standard can be generically approved, and this is under 9 CFR 412.2. Now, alternately, companies may request a blanket approval to use the disclosure option that they choose under the standard for all of their bioengineered foods produced at that establishment. A blanket approval refers to situations where the submission and approval of one label application covers an entire line of products without having to submit each label individually. And we refer to our compliance guide, which is entitled FSS Compliance Guideline for Label Approval for additional information on requests for blanket label approval. Next slide.
Hey, John, I just want to make sure that we have time to do Martha's presentation as well.
Okay, I'll hurry up to this then. Okay, just historically, just want to point out that in term, FSS has allowed the use of the term genetically modified organism, GMO products for label and labeling only if the name of the third-party certifying organization contains these terms. A few years ago, we reconsidered this position and has allowed and will continue to allow the use of the term genetically modified organism or GMO in negative claims, providing that the label or labeling is otherwise truthful and not misleading. Next slide. And here are some examples of claims wording that can be seen on this slide. Next slide. Okay, because FSS does not have the ability to independently verify negative claims for ingredients or feed, we require establishments that make these claims to comply with standards established by third-party certifying organizations. And we reviewed their third-party certifier standards and require that the standards be publicly available on a website. Now, the National Organic Program is one example of a third-party certifying organization for negative claims. As previously mentioned, we do not limit claims to those consistent with AMS definition established in the National Bioengineered Food Disclosure Act. A third-party certifier can utilize either AMS's definition or the Food and Drug Administration definition of modern biotechnology. Next slide. I'm going to skip over this slide for sake of time because all this information is available on our website. Next slide. And we have published a compliance guide called, next slide, please. I'll go back one. Go back again. Okay, yeah. We have published a compliance policy guideline entitled Statements that the bioengineered or genetically modified ingredients or animal feeds were not used in meat, poultry, and egg products. We sought comments on this guideline as a part of efforts to continuously assess and improve the effectiveness of policy documents, and this contains much of the information that one would need in terms of negative bioengineering claims. Next slide. And for the sake of time, that policy document contains most of this information here regarding negative claims and wording that would appear on the label. Next slide. And just in summary, any negative claim made on a single ingredient meat, poultry, and egg product must specify that the claim is referring to the feed because these products do not contain multiple ingredients. For example, derived from beef, fed, no GMO feed. FSS does not consider animals to be bioengineered if they consume feed produced from, containing, or consisting of bioengineered substances. And labeling of multi-ingredient products may include a negative claim that refers to animal feed or may bear a negative claim specific to non-meat or poultry ingredients used, for example, contains no GMO ingredients. Next slide. These are just a couple examples of negative claims. And go to the next slide, please. And I won't go through these for the sake of time. If there are, if there not any next slide. If there's not anything else, I'll conclude here.
Okay, so we can go to the next slide, please. So I already talked about the outline of my presentation today. Next slide, please. And this is the overview, the structure that we have in the biotechnology regulatory services. We are part of the USDA, the Department of Agriculture, that has around 100,000 employees. USDA is subdivided into 29 agencies. One of them is APHIS, that we have 8,000 employees. And then within APHIS, we have the Biotechnology Regulatory Service Program. And within BRS, we have the Biotechnology Risk Analysis Program. That's a program that I am part of, that it has around 30 employees. And the work that we do in BRS is coordinated across two different programs, BRAP, and then we also have the Regulatory Operations Programs, in addition to other 4 service groups that we have from the Office of the Deputy Administrator. Next slide, please. And this is the organization chart that we have in BRAP. What we have at the top is the director of our program. Then our BRAP is subdivided into 3 different branches. The first one is the Plant Pest Protectants branch, which I am branch chief, and we have 19 members. Then we have the Plant Evaluation branch, which is managed by Suma Chakrabarty, and it has 8 members. And then we have the Plants branch, which is managed by Deshri Sang, and his branch has 9 members. And the role of BRAP is to address potential plant pest risk and environmental impacts of modified organisms on the human environment. And we do so by conducting different risk assessments and environmental assessments of this product. My branch, the Plant Pest Protectants branch, specializes on performing risk assessments of modified microbes. I have been in this position for close to a year now, and I'm very lucky to be able to interact with such a group of talented scientists. We have scientists in diverse fields that are experts in plant pathology, microbiology, botany, entomology, virology, molecular biology, plant breeding, biochemistry, and others. Next slide, please. And the role that we have in BRS is to protect the US agriculture and agriculturally important natural resources by regulating organisms developed using engineering that may pose a plant pest risk. And we do so under the authority of the Plant Protection Act of 2000. Next slide, please. So let's talk a little bit about our revised biotech regulations that they were published in the Federal Register in 2020, and they replaced the legacy of the CFR Part 340. And this revised biotech governs the import, interstate movement, and environmental release of certain plants, insects, and microorganisms. And it represents really a new approach for US regulation of plants. These regulations were developed based on 3 decades of experience and advances in science and technology. And it better focuses the regulatory resources on areas of plausible risk. Next slide, please. And with our revised regs, we have 3 regulatory processes in place. The first one is the exemption and confirmation, and is to determine if a plant meets the criteria for an exemption with the option for requesting confirmation of plant exempt status. And I want to highlight that this exemption is only for plants, for modified plants. The second process that we have in place is the RSR, or the Regulatory Status Review, and is to determine if a plant developed using genetic engineering poses or not a plant pest risk. Also to highlight that this process is only for plants, for modified plants. And the third process that we have in place is the permitting process. This one is for plants and modified microbes, and is to apply for a permit for a regulated organism that does not undergo or pass the RSR process. Next slide, please. And in this slide, I wanted to summarize what are the organisms that are regulated by BRS. We have the plants, unless they are exempt from regulations. Then we have plant pests. Then moving into biological control, if they could pose a plant pest risk. Then if it's not a plant, but it has received DNA from a pest, And this DNA can produce an infectious agent or a compound that can induce plant disease. And the fifth and last group is PMPI plants, that stands for plant-made pharmaceutical or industrial compounds. Next slide, please. So I'm going to be jumping into the regulations that we have in place for modified microbes. So I thought it was a good idea to start with a few definitions of terms that I will be using. First, genetic engineering. That is a techniques that use recombinant, synthesize, or amplify nucleic acids to modify or create a genome. Then moving into the definition of plant pest, that it is any living stage of a protozoan, non-human animal, parasitic plant, bacterium, fungus, virus, infectious agent, or other pathogen, or any article similar to, allied to, with any of the foregoing that can directly or indirectly injure, cause damage to, or cause disease in any plant or plant product. And the last definition I want to go through is the plant pest risk. That is the potential for direct or indirect injury to, damage to, or disease in any plant or plant resulting from introducing or disseminating a plant pest, or the potential for exacerbating the impact of plant pests. Next slide, please. So in this slide, we have a summary of what are the modified microbes that are regulated by BRS. We regulate a modified microbe, if first meets the definition of a plant pest, or second, it has received DNA from a plant pest, and the DNA that has been transformed with is capable of producing an infectious agent, or is going to encode or synthesize a compound that causes plant disease. And the third type of modified microbe that we regulate is if it acts as a biological control agent to control plant pests and could pose a plant pest risk. Next slide, please. So this is one of the most common questions that we receive in my branch about the timelines that we have for permit applications for the approval or denial of a permit. And what you have here is the APHIS e-file website, the website where you submit that application, that I'm going to share the link at the end of my presentation. So the timeline that we have for approval or denying a permit is 45 days for an interstate movement and importation application after we have received a completed permit application package. For releases, the timeline is 120 days after we have received a sufficiently completed permit application. So then let's jump into what are the requirements that we have in BRS for applying for a permit. Next slide, please. Okay, so in the next 3 slides, I summarize what are the application requirements that we have for interstate movement and for release applications. So in this first slide, we have for interstate movement and importation permits. We need the name and contact information of the responsible person. We need to know about what's the organism that the modified microbe that you're applying for. It has to be at the species level. We need information of the intended trait, what has been the modification method that has been used. We also need information about the construct. We need to know about the components, the donors. We need to know the species of the organisms that have been used to donate this genetic material. And for the components, we also need to have a detailed description of the functions. Further, we need to have information about the origin and destination of the modified organism, including the address of the person who will be receiving and who will be sending the modified microbes. And in the next 2 slides, I want to talk about environmental releases. Next slide, please. So I want to highlight that the requirements that I have here are on top of the requirements that I went through in my previous slide. So for environmental releases, we need to know about the location where the modified microbes are going to be released, including the area, the GPS coordinates, the address, the land use history at the site where it's going to be released, but at the same time, the history of the adjacent areas. We need to know about the purpose for introducing this modified microbe, including a description of the proposed experimental design. And lastly, for this slide, I want to know, we need to have information for the confinement protocols that will be taking place. We need to have a description of the actions that will be taken to maintain that the modified microbes are going to be maintained at the release site, and what are going to be the protocols that will be in place to be able to prevent the spread and the persistence of this modified microbe after the termination of the field trial. Next slide, please. Continuing about the requirements for environmental releases, we need information of the intended trait and the genotype of the intended trait, including the diagnostic test that has been developed to differentiate between the wild type and the modified organism. And this diagnostic test is going to be used during the monitoring phase of the field site to be able to make sure that it has been confined in the site, the modified microbes. For the monitoring, we need a description of the monitoring duration and also the frequency to ensure that the microbes will not persist in the environment. We then need information about the final disposition for release, including the description of the method to be used for the final devitalization. Also to highlight that if we issue this release application permit, APHIS will be inspecting to assess the compliance with the permitting condition, and we will require the maintenance and submission of certain records. And in our revised regs, we have a couple of exemptions for modified microbes for permitting requirements. Next slide, please. And these are the, excuse me, the two exemptions that we have. First, we have for modified disarmed Agrobacterium species, and a permit for the importation or interstate movement is not required for any disarmed agro species. And we have to make sure that it's provided that it's moved in a secure shipment, that the modified genetic material is stably integrated to the genome, and that the modified material does not include the complete infectious genome of a plant pest. There's an option for stakeholders to request a letter of no permit required from BRS for importation for Agrobacterium species. If you need one, please do not hesitate to send me an email, and I'm going to have my email address at the end of the presentation. The next exemption that we have for permitting requirements is for certain microbial pesticides, and a permit is not required for the movement of any modified microbe product that is currently registered with the EPA as a microbial pesticide, highlighting that this microorganism has to be not a plant pest. Next slide, please. So I thought it would be a good idea to put a couple of examples together about when a permit is required or not from BRS for the movement of a modified microbe. And what I have here, this slide is a very common query that we received in my branch when an organism has been transformed with GFP, for example. So here we have the organism as Bacillus subtilis, which is a soil bacterium. The donor organism is jellyfish. The construct is GFP, and the function of GFP is to use it as a fluorescent marker. So in this case, because Bacillus subtilis is not a plant pest and the donor organism is not a plant pest that can induce disease, then therefore, for the movement of this specific case, a BRS permit is not required. Let's see when one is required from our group. Next slide, please. And here we have an example of when a permit is required. We have as the organism, genus, species Fusarium verticilloides, which is a fungal pathogen. The donor organism is Aspergillus needleland. The control component is a zinc finger transcription factor. And the function of this transcription factor is to increase asexual development. In this case, because Fusarium verticilloides is a plant pest, the applicant will need a permit from BRS. In the next slide, I want to share with you some of the ongoing projects that I have in my branch with modified microbes. Next slide, please. So first, we are in the process of developing a microbial regulation FAQ document. So we have gone through the biotech queries for modified microbes that we have received in the last 3 years. We have compiled all these hundreds of queries, organized them in different topics, seeing which are the most frequently asked questions. And we are summarizing them and be able to put them out in our website for stakeholders to be able to have access to it. Next, this should be coming in our box, yes. And the other project that I'm working with a few of my team members, we're in the process of developing a microbial guidance document with information requirements and process for submitting microbial permit applications for movement, importation, but also for environmental releases. And we will share this document and invite the public for comment. Next slide, please. And in this last slide, I wanted just to share some links and information for our stakeholders. We encourage our stakeholders to contact us for any type of pre-consultations. We are happy to meet with you. We meet on a weekly basis with the stakeholders to address any questions that they may have to know if a modified microbe requires a permit from us or not. And you have here my contact information and my email, so please send me an email if you would like to set up a consultation meeting. Next, I have here the VRS site website if you want to know more about our program. The next biotech query, this is an email inbox that we have in VRS. I think that is super helpful, where applicants can submit a question about a modified plant or a microbe or an insect to know if they require a permit from us or not. So we go through these queries, and within a couple of days, we will acknowledge the receipt of this email, try to help you, and maybe if we need more information to be able to address the questions, we will let you know. The next link is about the revised biotech regulations, where you can find more information about them. And the last one is the site for e-file, where you can submit a permit application. And with that, I want to thank you all for your attention. Next slide, please. And I don't know if we're going to have time for questions or not. Thanks.